Hi, everybody, and welcome to today's lecture, where we will be focusing on um, our constant allele frequency and using an equation called Hardy-Weinberg equilibrium to calculate the frequency of medical conditions or chances to develop disease or being carriers in um, single gene-related disease. So for us to first understand the concept of Hardy-Weinberg equation, we need to understand some of the terminology that helps us understand the concept a little bit better. So some of these words we already described in our previous lectures, so it might be a review, but I will go over them one more time. So one of the first thing that we have is what we call populations. So a very interesting fact about population is the fact that they have the capacity to interbreed with each other. That means that they have the capacity to create your next generation offspring, and those offspring also have the ability to create the future offsprings. So they have to be of the same species and within the same geographical area. The very important fact about the genetic um, populations and having the same species is as they move away from certain geographical areas, depending on the environment, natural selection, gene flow, mutation, etc., it would lead to creation of the new species, and those species cannot interbreed. So population have to be the same species. Now, when we talk about gene pool, the concept of gene pool is when you look at all the alleles that are present within a population. So if you have thousands of members and you have variation within the alleles, all of that collected together will be considered your gene pool. Now, since we're talking about population and the concept of genetics, when we talk about population genetics, is basically looking and studying anything that's related to a population's gene pool, as well as genetics and mutations that may happen in the allele and how those allele may be varying over time. And last thing, one of the most interesting things is the concept of gene flow. Gene flow is basically when your allele moves between the populations as a result of either migrations or mating. So as the name implies, gene flow of genes from one location to another. And again, the most common reasons when you would have this variation in that population in your gene pool is due to migrations and mating. And we'll go into more detail about a specific type of mating called non-random mating in just a few minutes. Now, when we're talking about the concept of Hardy-Weinberg equation, one of the first things that we want to kind of understand is the concept of frequencies. So frequencies is literally thinking about all the genes in the populations and considering how frequently or how often they happen within our population. So when we looking at this, we're talking about both the presence of alleles, development of the genotype, and how those genotypes basically represent themselves on that individual as a phenotype. So we're looking at alleles, again, the variation in the genetic information, what genotype they create, and how those genotype create your phenotype, the physical characteristic or something that is, again, represented. Now, and most of these are frequencies and changes that may happen to those frequencies as a result of mutations. It could be as a result of repeated DNA sequences or mutation, I should say a mutation and repeated DNA sequences are going to be the most common things that can affect the gene population and basically cause variation in your allele, genotype, and phenotype. Now, in this presentation, we're only talking about genes that are controlled by a single allele. So these are single gene traits. We talked about them before versus when we talk about complex traits, when you have multiple genes and epigenetics actually affecting the presentation of that gene or the presentation of allele, genotype, and phenotype in the population. So we're talking about single gene, and the equation that we're using is Hartley-Weinberg equation. Now, as I mentioned, this situation will become more complex as we're talking about complex traits where there are multiple alleles or when you have multiple genes controlling a phenotype, a specific phenotype. Now, when we're talking about frequencies, another thing that we want to kind of think about is depending on where the individual is growing, depending on the interbreeding in a population, we're talking about human population in this case, how does that affect a frequency of certain single gene disease? So again, we're talking about if there is a mutation of any sort in the allele, what are the chances of that person either being the carrier or have the actual disease presented on them. So depending on the population, these frequencies may be different. For instance, there's an example of the medical condition called cystic fibrosis. And we talked about cystic fibrosis in detail in our first few lectures. Basically, it's the one that builds up mucus in the lungs and affect multiple systems. And cystic fibrosis will be more common in Caucasian children. And this is something we'll look at frequencies later on. But if we're looking at the African American population, again, looking at different population, we're looking at sickle cell anemia being the main, I guess, one of the main medical conditions that will affect African American at a higher rate compared to other races. So frequency of singles gene disease are different in population, which is what we're trying to say. And there is some examples here for you to kind of show you one of the medical condition called phenylketonuria or PKU, which is where your body is unable to break down very specific amino acid called phenylalanine. And we have how each of the population have different frequency of them being infected by this single gene disease. For instance, if you're looking at Chinese population, they are relatively, I guess, less affected by this. Japanese are really not affected. So we have 1 in 119,000 being affected by this. But the two numbers that are kind of, I guess, concerning is Turks being affected because 1 in every 2,600, and Irish, Scottish, and Yemeni Jews are 1 in every 5,000 being affected. So again, you can see populations being affected by these medical conditions at a different rate. So when we go and do kind of genetic counseling, some of this information will basically be used. Your background in terms of your race can help you identify what is your risk of developing certain medical conditions, or if you are carrier for that, possibly, and may pass that to your child. Now, when we're talking about the concept of evolution, so evolution is basically changes in the population over time. We can talk about it in terms of microevolution. As the name implies, it means small changes over small changes in the population versus macroevolution, which is a much larger changes. So when we talk about microevolution, what can cause or affect these changes in the population over small changes in a population. So one of the things is called non-random mating. Another thing is migration, genetic drift, mutation, and natural selection. So again, these are microevolution that slightly changes the allele, the variation in the gene, and therefore affect the genotype and eventually affecting the phenotype. So migration, I think, is self-explanatory, is when a specific number of population move to a different area where you have different resources, and that can trigger certain characteristics to be developed versus the other one. Mutation, we have talked about it in detail, so I'm not going to go over it again. but any sort of mutation that basically causes changes in the DNA sequence and effectively the genotype and phenotype. And the other three that I want to kind of briefly go over is the non-random mating, the genetic drift, and natural selection. So natural selection is one of the most important things that allow that microevolution to happen. So natural selection is when you have an organism that has a better chance of surviving in the environment compared to the same species, but may have characteristics that limit their survival within that environment. So the one that's basically surviving can reproduce, create the next offspring, and pass down those good, quote unquote, good characteristics to the next generation. And you can see that by looking at the picture next to on the side. We basically have the same species of beetles, some of them green beetles, some of them are orange beetles. So based on the color and the environment, the green beetles are much easier to be picked by the predator, in this case, your crow, which means now the orange ones are going to be more safe and therefore start to reproduce. And as a result, the number of the orange ones are going to increase over time, causing most of the species, if not all, representing the same characteristic of color, which is orange, protecting them against the predators. Again, you're naturally being selected against certain characteristics based on the environments, based on predators, based on the amount of water, food, etc., resources. Non-random mating is another component that leads to microevolution over time. If you're thinking about a mating process, most of the mating process, you think about it as random. So what does that imply? So here is an example of a random mate. You have a brown beetle, and that brown beetle can mate with either kind of light brown color, the same color as it, or a very light color, almost yellowish color. So that's random mating, which means the chance of this beetle mating with those three in the middle is completely random. 1 in 3 for each of them. Non-random mating, it means that the beetle that you have a preference for a specific characteristics, and that could be depending on the environment or the physical characteristics can make it superior in that environment. So in this case, it might be the color, so making this non-random. So when the female beetle, for instance, is looking for a mate, it's looking for something that is the same color as hers and basically ignoring these two on either side. So you're choosing it based on that better physical characteristics. So those traits are going to be passed on more often, causing microevolution in the population. And the last one that I wanted to describe with you, and I show you kind of in the visual, help you visualize this, is the concept of genetic drift. So the genetic drift is basically where the population or the frequency of allele in a population completely changes by random. Again, example of this, you start with a population of beetles. You have the same ratio for the brown versus the green one. So it's 3-3 or 1-1. But by random chance, something comes in and knocks out 2 of the green ones. So now your population is 1 compared to 3. Now, because you have 3 of the brown ones, they have a higher chance of reproducing and passing that characteristic. So therefore, you're going to end up with more brown beetles over a long period of time versus the green ones. Again, this happens completely randomly. Let's say a tornado comes in and takes half of the population away, and most of those were green. It doesn't have to be a specific changes in the populations, anything that literally changes by random chance the frequency of a specific allele in the population. Now, when there is enough microevolution that happens and there are enough changes in your organism where the two fertile organism of the population cannot produce a fertile spring offspring, then you have created a new form of a species. So basically, if you're taking away the ability to reproduce, you have created a new species. Um, Now, macroevolution, on the other hand, as the name implies, macro means large. These are changes that are great enough to result in speciations in terms of, again, macroevolution. Changes that are really large that causes two populations to be pulled apart from each other. Now, as I keep saying, microevolutions are small changes over time, microevolution, or a few big changes that greatly affect the phenotype, which is typically implying macroevolution. So microevolution, here's an example. Microevolutions are changes such as variants in a specific gene called FOXP2 gene that enable humans to speak. So that's microevolution. It happens over a long period of time versus larger scale changes, which is macroevolution, separated us from their closest primate relative, which are basically our major primates, such as gorillas and monkeys, or apes, I should say. So macro versus micro. Now we talked about alleles, we talked about frequencies, we talked about how population is shifting to create a new species based on microevolution or macroevolution. Now we're going to focus on looking at a population and basically by looking at the population, say what is the chance of a specific allele being present in that population, and therefore figuring out what is the frequency of each genotype and therefore each phenotype. Again, physical characteristics versus the combination of the allele within the population. So the way we do that is pretty simple calculation. For each of the allele frequency, we take the number of individuals within the species and then divide that by the total number of populations. Now, allele frequency, again, will affect the genotype frequency, and there is the genotype that are either homozygous or heterozygous. So kind of, again, think about the population. You can have homozygous dominant, homozygous recessive, or heterozygous, in this case, heterozygous dominant, which is the only option you have. Now, I have mentioned Hardy-Weinberg equation a couple of times already in the lecture, so we are using that information to, again, figuring out how frequent an allele is in the population, and therefore what is the frequency of a specific genotype and a specific phenotype. Now, this equation was developed in 1908 by a mathematician and a German physician. So again, kind of collaboration between the sciences, which is really cool. And it's using the concept of algebra to explain and figure out or predict what is the frequency of the future population would look like in terms of, again, phenotype and genotype. And it is also this equation is also disproving that the fact that because you have a dominant trait, that dominant trait will eventually become more common because it's dominant, and then the recessive trait, as time passes by, becomes less and less common. So as we look at this equation, you will notice that frequency of dominant and recessive, the ratio is retained going from one population to another. Just because something is recessive, it does not mean it decreases the frequency over time. Now, for this equation, for us to work, we need to assign specific letters and then use the equation to basically solve for each of the frequency and genotype and phenotypic variation within the population. So we're going to use the letter P and Q P and Q represents a variation in the allele. So P is going to represent one of the alleles, and then Q is going to represent the second alleles. And we're going to say P plus Q is equal to 1. Again, this is the concept of that equation, allowing the consistency within the population. Now, if we add all of the allele frequency together, that's also equal to 1. So if we have P squared plus 2Q squared plus Q, this is, again, genotype, that's equal to 1. So all of the genotype, again, together would be P squares and Q squares. Now, frequency for the homozygous are represented as P square and Q square, and for heterozygous, it would be 2PQ, which is what we have on the equation. So again, here is your equation. P square plus 2PQ plus Q2 or Q square is equal to 1. So homozygous dominant, homozygous recessive, and in the middle you will have the heterozygous characteristics and genotype. So again, we're using the Punnett square, applying the same concept in terms of frequency. So right here we have a heterozygous for both sperm and egg that are contributing their genetic information. And this is where that P square 2PQ Q square comes from. This is your P square. This is your Q square, and these two together is your 2PQ. To kind of give you an idea where that equation comes in. And again, all of this will be equal to 1. So if we have any information, either just simply the P or the Q, we can use the equation of P plus Q equal to 1, and then P squared plus 2PQ plus Q squared equal to 1 to solve for the frequency of each genotype and phenotype. Here's an example of what that looks like. So it says, given the P frequency of D, so P frequency of the D, which is associated with the characteristic represented here, being a normal length fingers, you have a frequency of 0.7, and then the rest would be basically for the Q, right? Dominant and recessive. So frequency for the recessive version of the gene, which gives you the short middle finger, that would be 0.3. And again, if you kind of looking at these numbers, if I add 0.3 and 0.7, that's equal to 1, which is simply P plus Q equal to 1. So if you're using the Hardy-Weinberg equation, as well as combining that with the concept of the Punnett square, we can deduce that D, so if you have a homozygous meeting with a homozygous, right? So capital D, lowercase d, capital D, lowercase d. Both of these are 0.7, and the lowercase d or q would be 0.3. All you need to do is now multiply these numbers. So dd, which is going to be p squared, 0.7 times 0.7 is equal to 0.49. dd, which is lowercase dd, is q squared, which is the recessive gene. Again, that gives you q squared, and you have 0.3 and 0.3 combined together. So that's equal to 0.09. And then 2PQ, which is the heterozygous one combined together, it's 0.3 times 0.7. So you have 0.21 plus another 0.21, 0.21. So that would be 0.42. So that would be, again, 2PQ. And here are your three numbers. That is represented down here. 0.42 for heterozygous, 0.49 for homozygous, and then 0.09 for the homozygous recessive version of the gene. Now, again, I can use this equation, exactly what I did, to figure out, based on the genotype of the parents, whatever the combination is, what is the frequency of them passing that characteristic to their kids. So for instance, to just kind of show you this, passing out the characteristics of sorry, if you have a male that is capital D, and then capital D, what's the chance of them having any variation within the genotype? And you notice that it's always going to be capital D. And then again, looking at them, if you have a homozygous dominant plus a homozygous, heterozygous, what's the chance of them creating the a specific allele? So it gives you the ratio. And at the end, if you add all of these together, right, the variation in DD or homozygous dominant, homozygous recessive, and heterozygous, you still add all of them to be equal to 1. So it doesn't matter how we talk about this, what is the different chance of combinations, it's always equal to 1, because it's always going to be the variation of the alleles, and then to form homozygous recessive, homozygous dominant, or the heterozygous version of the gene. Now, this is very important for us to remember when we talk about the Hardy-Weinberg equation. They can be used for simple or single gene disease also, to figure out if somebody is a carrier of a disease and what is the chance of that carrier passing that medical condition to their children, or having the child born with the actual disease. So let's take a look at, again, examples of this Hardy-Weinberg in a little bit more detail. I mentioned that earlier, that if you know either the P and Q, you can actually solve for the frequency of the genotype as well as the chance of you having that medical condition, because of the following equation. P plus Q is always equal to 1. And again, it is for autosomal recessive disease. The homozygous recessive class is used to determine the frequency of the allele in the population. Because that's the easiest way, since that is the reason you will develop that medical condition. And then phenotype indicates its genotype. So phenotype Q2 would basically, Q2, Q squared is basically a determining factor for the specific genotype that you will get. So let's solve a question and see what that answer looks like for us to figure out the genotype, and again, the chance of us developing a specific medical condition. In this case, we're focusing on cystic fibrosis that typically affect the Caucasian population. So cystic fibrosis, as I mentioned earlier, is more common in the Caucasian population, and you may not develop the symptoms of the disease, but rather be a carrier. So you want to consider that, which is basically means that you're a heterozygous. So there is the same piece of information. It says that autosomal recessive characteristic, which basically means Q2, is 1 in 2,000 people. So for us to figure out the frequency, all we need to do, as we learned earlier, we learn about the equation, take the number of that specific individuals with that medical condition and divide it in the total population. So 1 in every 2,000 gives us 0.0005. Now notice that this is representation of Q squared. So it means this is Q times Q, right? This is homozygous dominant, homozygous recessive, right? Because that's what we're looking at. So now I need to just figure out what's my Q. And all I need to do is calculate the square root of 0.005, and that's equal to 0.022. Now, because I know my Q, right? I know my Q. All I need to do is says P plus Q is equal to 1. Replace that, like 0.022 equal to 1 plus P. So solve for P, which is 1 subtracted from 0.022, which gives us 0.978. And that would be my P, which is what you see calculated here. So P is equal to 0.987. So I have figured out what is the chance of the allele, the dominant allele being present, 0.978. What is the chance of the Q being present? And it is 0.022. Now all I need to figure out is what is the heterozygous dominant allele or combination, more specifically genotype, can be present. And all I need to do is do 2PQ, which is 2 times 0.97 times 0.022, and that's 0.043, which means 1 in every 23. So what are all these numbers mean again? So based on the fact that cystic fibrosis is an autosomal recessive disease, 0.22% of 0.022% of population are basically, or that translates to 1 in every 2,000, will basically have the cystic fibrosis. That just basically means they have the medical condition. 1 in every 23 are going to be carriers. And then you need to do the calculation here. I haven't done that, but 0.978, that translates to the frequency of the population basically being normal. So regardless of how many times you do this, one of the main things that I mentioned is that the typical understanding was that because I have an autosomal recessive disease, the concept of recessive is going to decrease in frequency over time versus something that's dominant is going to increase over time. And we can see by following this equation that this is not going to be the case. You will always have a dominant, you will always have the recessive. It depends on how that gets passed down to the next generation as either the person being a normal person, a homozygous recessive that does have that medical condition, or a heterozygous where they are identified as a carrier. Now, if again doing the cystic fibrosis based on the populations, African American, there is a chance of 1 in 66 to have the medical condition. Sorry, not to have the medical, to be the carriers for the disease. Asian Americans are relatively low, so 1 in 150. And Caucasian and Hispanics are on the higher side when you have 1 in 23 and 1 in 46 being the carriers of the condition respectively. Now, what is the probability that 2 unrelated Caucasians will have an affected child? Again, thinking about the concept of cystic fibrosis, what if you have 2 people in a population, they're not related, what is the chance of it being passed on to their child? So all we need to do now is figuring out that if they are carriers for that medical condition. If they are carriers, assuming they're both carriers, which is we figured out is 1 in 23 for Caucasians, and you can change that based on the population. So 1 in 23 chance for the mother to be a carrier, 1 in 23 chance for the father to be carrier. If you multiply that together, there is 1 in 4 chances for the, sorry, not 1 in 4 chances. 1 in 29 times 1 in 29 times 1/4 of that population having the QS square. So it's 1 in 2,116 to have that medical condition. So again, probability is all about multiplication, because the new factor that you add, you're going to multiply that by the the frequencies that you have. So if you have each parent being a carrier, that goes up to one out of 590, 529. And then because the allele for the Q square is 1 out of every 4, right? Think about your Punnett square. P square, Q square, uh, PQ, PQ. So to having a Q square is 1 out of 4 chances. So you take the probability of the genotype of the parents times the probability of that being passed down to the child, multiply those together, and you get 1 out of every 2,116 child may be a fully affected individual with that autosomal recessive condition. Now, we can, again, based on what I just showed you, we can calculate with accuracy what is the effect of the Q, sorry, not the effect, I should say, the frequency of each of the genes and how it gets passed down to the next generations. Now, because these single gene diseases are so rare, it's typically the Q square, which is the frequency of the person having that medical condition is a very small number. Just by looking at the Hartley-Weinberg equation, as we just saw, it's a very low frequency compared to the overall population. Because of that, that means if we have P plus Q equal to 1, P is going to be closest to the 1 because this number is going to be very small. So P is going to be almost equal to 1. That's one thing. So that means your homozygous, if I can spell, homozygous dominant characteristic, which is P squared, would be equal to 1 almost. Now, the chance of having a heterozygous, I can't spell either, heterozygous dominant, which is 2PQ, is basically 2 times a number that's very close to 1, which basically means your Q is typically twice the amount of your Q. So Q is always, I should say it backward. Your frequency of the 2PQ is almost always twice as much as your Q, because the P is always close to 1, so it's equal to 1. So whatever number you multiply by 1, you end up with the same number. And because you have 2PQ is 2 times whatever number your Q is, so your PQ or your heterozygous characteristic is going to be double the Q number. So that's about it. I would recommend for you guys to look online to search for some questions about the Hartley-Weinberg equation, so you can get some practice, and that will also help you guys get better prepared for your exam. But overall, the most important thing that you want to focus on for this is, one, look at your numbers, make sure that you understand where those numbers come in, be able to distinguish between the frequency of the genotype versus the phenotype characteristic developed as a result. So P plus Q is equal 1, and Q square plus P square plus 2PQ is equal to 1. And then making sure that you know how to plug in those numbers and calculate for the missing component. And lastly, be able to describe the main reasons of development of microevolution, providing examples of that, and then looking at some of the main vocabularies that I discussed at the beginning of the lecture. Thank you guys, and best of luck.